Okay, so in this video, we're going to look at how to identify organelles from diagrams. Okay, so I suppose most of you would be pretty used to this kind of slightly more cartoonish drawings, which usually are much more easy to recognize. Right. However, in the A level, very often you will be given something more realistic, right? The electron micrograph, right? A photo taken with the electron microscope. Okay, so this is when it makes it much harder to identify the different organelles. So in this video, right, I'll be going through some tips which may help you to identify the organelles given the electron micrograph. But do take note that in real life, right, organelles obviously can appear differently depending on how the photographs are taken, right, the angle that it is taken, the lighting, and so on and so forth, right? So while I'm giving you some tips, right, they are not like absolute, yeah. So they're just there to help you to start thinking or try to identify the organelles, right? It's not like this magic bullet where you just follow this list of characteristics and therefore you surely can identify the organelles. So it takes a lot of practices, okay, which obviously you can find questions to practice, right, with our tutorial, the EMCQs, and so on. Right, so I'm just going to go through some more common distinguishing features for individual organelles to help, right, at least give us some hints when we are identifying the organelles. Right, so let's start off with the first one. For those who are quicker, you will be able to recognize that this is the nucleus or 3 nuclei, right? So what are some distinguishing features visually, right, in the electron micrographs? Right, so you will usually see that the coloration of the nucleus bound by the nuclear envelope, right? Inside the nuclear envelope, the coloration would be of some darker regions, some lighter regions. They are in patches, right? So the nucleus in the center, right? That one is very obvious because the dark patches are really quite dark. But for the one on the left and on the right, even though the distinction between the dark and light are not so obvious, right? But still you can see, right? It's not a uniform color, right? There are certain patches that are darker and lighter, and they are all like mixed together, right? So dark, light, very irregular kind of coloration. Right, so that will give you a hint that this may be the nucleus. Obviously, you can look at the size, right? If you can see the whole cell, if the electron micrograph shows the whole cell or a large part of the cell, then you can obviously compare the size of the nucleus to the size of the cell, right? Because nucleus is the largest organelle, usually. Okay. And usually, right, within the nucleus, there would be one or more very, very dark regions, even darker than the dark patches that I mentioned just now, yeah? So the very, very dark region, right, would be the nucleolus, right? So if you can identify the nucleolus, then obviously the bigger organelle with the membrane would be the nucleus, right? Just take note that there might be more than one nucleolus, So in the 3 nuclei that I'm showing, all of them only have one nucleus, rather only have one nucleolus, right? But do take note that some nucleus can have more than one nucleolus or nuclei, yeah. And for certain diagrams of the nucleus, which happens to be very, very clear, you might be able to see the double membrane, like the one on the left, the nucleus on the left. You can actually see 2 layers of the membrane, right, on the nuclear envelope. Yeah, so that would be very lucky that you have such a clear diagram. Yeah, so that would give you more confidence that this is a nucleus, because not all organelles have a double membrane. And sometimes, right, if it's again a very clear diagram, you might be able to see some holes, right, some nuclear pores, yeah, on the nuclear envelope. but I don't think we can see that in any of my 3 nuclei here lah. Okay, so really the more common kind of distinguishing feature would be the irregular patches of coloration, and usually there should be the nucleolus also. Not a given, they may not have, but usually there will be either one or a few. Yeah, so those are more common. Next, okay, this would be the mitochondria, 4 of them actually. So the most telltale, the most obvious distinguishing features of mitochondria would be lines going across the organelle, right? So in all 3 diagrams, or 4 mitochondria, right, because the middle diagram has 2, you can see the lines going across, right? So the lines would be the folding of the inner membrane or the cristae, and if you can see such lines, right, going across the organelle, then quite confidently you can identify that this is the mitochondria. Okay, so far, right, we have gone through nucleus and mitochondria. You realize I did not talk about the shape, yeah, because some of your seniors would say that, right, the nucleus usually is round, the mitochondria usually is bean shaped. But right, for the mitochondria you can see here, they are not all bean shaped, right? So again, the shape, right, is not absolute, right? It really depends on the angle that the photograph is taken, right? Whether the cell is distorted. Sometimes when the cells are packed into tissues, some of the cells shape and organelles shape might be distorted, and other reasons. So really don't base it on the shape. Yeah, it might give you a clue, but you need to base your confirmation on some of these distinguishing features which are not present in other organelles. Okay, so back to the mitochondria. If you see the cristae, then you can be more confident to conclude this must be mitochondria. Okay, then move on to the chloroplast. Right, for the chloroplast, the distinguishing features would be that you see a lot of lines or membranes going across the organelle, a lot more than the mitochondria, right, if you compare this this slide with the previous slide, right? And the lines going across the organelle, right, are not uniform in color, right? Just how the mitochondria, they're just like those sparse lines that don't really have different coloration. In the chloroplast, usually we'll see different colorations within the organelle as well, and the different coloration, the different patches of coloration, right, are more regular compared to the nucleus, right? Very often they are in rectangular blocks. Yeah, so all those are actually the granum or granular. Yeah, the more dense, with the overlapping stacking of thylakoid membrane. Yeah, so those are the granular, and they appear as rectangular or regular blocks of darker color. So when you see that, then usually you can be confident that this would be the chloroplast. Okay, another feature that not so confirming but may give you a hint is the size of the organelle. Again, if you can see the entire cell, you can do some comparison in terms of size. Chloroplast usually is the second largest organelle, right? It's much larger than the mitochondria. It's just slightly smaller than the nucleus. Yeah, so if you can compare sizes, then together with the coloration, the blocks of darker colors, then you can confidently conclude this should be the chloroplast. Okay, next is the rough endoplasmic reticulum. I'm just going to say ER for short, so rough ER. Right. The most obvious, most distinguishing feature would be the appearance of black dots, which are the ribosomes attached to the membranes of the rough ER. So once you see that, you see the dots, then confidently you can conclude that should be the rough ER. Another hint, right, would be the shape of the membrane, right? They are usually a bit more sheet-like, more parallel. So that would be the rough ER. Okay. Then since we're talking about ER, right, I just want to make maybe a comparison between the appearance of the rough and smooth ER, right? So you can see here the rough ER on the left, more sheet-like, more parallel, and you can see the ribosomes, right, the black dots. For the smooth ER, right, obviously you don't see ribosomes, and usually, right, smooth ER appears less sheet-like, less parallel. They appears like circles, like a lot of small circles grouped together, usually. Okay, so if you see that, and these circles are somewhat continuous, right, or nearby the rough ER, then you can more confidently conclude that this would be the smooth ER. Yep, so a bit more comparing the rough and smooth ER. Okay, now you can see the shape of the arrangement of the membrane more side by side here in this diagram. Sheet-like, parallel for the rough ER, more circles for the smooth ER. Another diagram showing you, right, those on the left are the smooth ER, those on the right are the rough ER. Okay, again, left smooth ER, right rough ER, so don't be confused by my heading on top, rough on the left. No, it's the smooth one on the left in the diagram. Yeah, rough ER with the ribosomes, the sheet-like parallel structures of membrane, right, rough ER on the right. Okay, so why do the two types of ERs appear a bit differently in terms of shape, right? That's I think this diagram is quite self-explanatory. So if you imagine you're looking from the top, we're looking at the rough and smooth ER, then obviously the rough ER are more sheet-like and parallel, and then the smooth ER are more like circularly in shape. Yeah, this diagram is found in the notes Yeah, so you can use that to remind yourself, how to identify the two. Okay, next we move on to the Golgi apparatus or Golgi bodies. Okay, I'll usually just call it Golgi apparatus, so I just stick to apparatus, okay, in this video. Also, the distinguishing features would be that the membranes are stacked together, right, and they are one stack on their own. They're not connected to anything else, right? Unlike the rough ER, which are continuous sheets, right? The smooth ERs, like a lot of circles that kind of touch one another. The stack of membranes in the Golgi, this one stack is like on its own, right? Surrounding it wouldn't have like something else touching it, usually. Okay? And obviously there's no ribosomes attached to the membrane, so you don't see the black dots. So again, you realize I don't talk about whether the membrane is curved, because again, whether the membrane is curved depends on the angle that the photo is taken. Like you look at the Golgi apparatus on the right, that is showing very clearly a curved pattern with the concave and convex faces, but the Golgi apparatus on the right looks more straight, right? They're not curved. So again, because it's taken from a different angle, so the curving obviously makes it easier for you to identify, but without curving doesn't mean that it is not going to be Golgi apparatus without the curving, because it's still a stacking of membrane, and it is a stack that is like independent, like on its own, so that would be still the Golgi apparatus on the left. Okay, so just wanted to put the two ERs and the Golgi together. Right, so on the left would be the rough ER, again straight, sheet-like, but with the ribosomes, right, black dots. The center one is the Golgi apparatus, and the one on the right would be the smooth ER. Okay, next up is the lysosome. Lysosome is a bit more problematic because they do appear quite differently. They can have different appearances. Usually they are darker, but that's not necessary, because I've seen lysosomes that are quite light before. It really depends on whether there are substances within the lysosomes that are being digested. If there are a lot of substances, they will be darker. If the lysosome somehow is not doing anything, not doing any digestion, then there is nothing inside, right? Then it's going to be lighter. So usually dark, but not necessarily, right? The dark coloration may be uniform. You can see on the left, right, the lysosomes are uniformly black, most of them, but the one in the center, the lysosomes in the center, are not uniform in terms of coloration. So don't let this confuse you with the nucleus. Obviously the sizes of the organelles are different. Lysosomes are much smaller, and you obviously don't see more than one nucleus in one cell, right? But you do see multiple lysosomes. Yeah, so the coloration is a hint, but not don't let the coloration confuse you with the other organelles. And if you see that the organelle is fusing with another organelle, like the one on the right, you can see the fusion of the lysosome with the mitochondrion, right? Then the one that is fusing with the mitochondrion must be a lysosome. So if you can see the fusion happening, then that is more obvious. Okay, then the centrioles, right? For the centrioles, usually they come as a pair. So when you see a pair of structure, you don't really see membrane, right? A pair of structure, usually rod-like, then that would be the centrioles. Sometimes it doesn't appear rod-like, it appears more ring-like, a circle, right? Like the one on the left, right? There are two pairs of centrioles, the diagram on the left, two pairs of centriole. One of them is more, for each pair, one is a bit more rod-like, the other one is more ring-like, a circle. Yeah, so sometimes it appears like that. Again, it depends on the angle that the photo is taken. Like the diagram on the right, both are rod-like. Yeah, this is one pair of centrioles. Yeah, but as long as you see a pair of structure, more or less rod-like, no membranes, then that would usually you can be a bit more confident that this would be the centrioles. All right. Okay, so just some pointers to help you. There are a lot of questions in the SLS for you to practice, and of course, you can go to the tutorial, the MCQs to practice as well.